Tomorrow you're going to be doing a lab on the animal cell and the plant cell, and the overarching question of this lab is, what are the differences between animal and plant cells? So in this investigation, you will compare animal cells, or your very own cheek cells, and plant cells, or onion cells. So some background information on the lab, or on cells in general, is that one of the first scientists to ever look at cells under a microscope, which is what you're going to be doing tomorrow, was an English scientist by the name of Robert Hooke. He viewed and described the appearance of cork under the microscope and decided to name the tiny box-like structures that he observed as cells, because they looked like and reminded him of the small chambers where monks lived. By the early part of the 19th century, it was accepted that all living things are composed of cells. Cells come in a variety of shapes and sizes, and they perform different functions. Although cells may appear outwardly different, they resemble each other because they share common structures, some of these called organelles. In this lab, we will look at two types of cells, a human cheek cell and an onion cell, and see how they are similar and how they are different. If you will look to the right of the page in the screen, you will see the materials that you will be using for the lab tomorrow. You will first have a microscope, you will have 2 slides, you will have your iPad, you'll have cover slips to put over the slides, you will have tweezers, pencils, the connector that connects the microscope to the iPad, flat toothpicks, onion tissue, iodine, and methylene blue stain. So, the onion cell procedure. What you're going to do first is set up your microscope. Then you're going to get a clean slide. Place a drop of water on the slide. Then take a piece of onion from the bowl in the front of the room. Obtain 1 layer of epidermal tissue. This is just the thinnest layer that you can get from an onion. Place the layer on the tissue on the slide and then add a small drop of iodine to the slide. You will then place a cover slip on the slide, slowly lowering it over the sample to avoid creating air bubbles. After you do this, you're going to place the slide on the stage of the microscope and view the slide under the low power. Once you have found an area with several good cells, you're going to switch to the high power. Remember to only use the fine adjustment to focus at high powers. For the next step, you can do 2 things. You can either draw the onion cell in the area below here, or you can take a picture of the onion cell and place it, insert the picture into your PDF. Once you do either of these 2 things, you're going to label the cell membrane, the cytoplasm, the cell wall, vacuole, the nuclear membrane, and the nucleus. When you're drawing these objects as seen under a microscope, it's really important to draw clearly and make distinct lines, and it says only use pencil, but you're going to use your iPad so you can use just the different colors that it offers, since you can erase it easily. If you take a picture, all you have to do is take the picture and then save it to your photos and insert it into that little section below. Make sure when you put the picture or the drawing there that you provide the name of the object and the power under which it was observed. So put onion cell and then whatever power you observed it under. When you're totally done with the onion cell, go ahead and clean the slide and dry it for the next group so that it is ready to go for next period. The next thing you're going to do is the cheek cell procedure. So you're going to put a drop of methylene blue on the slide. Make sure that you are very careful with this because it can stain clothes and it can stain your skin, so be really careful with this. Then you're gently going to scrape the inside of your cheek with the flat side of a toothpick, and make sure you scrape lightly so you don't cut yourself. And then you're going to stir the end of the toothpick into the stain and throw the toothpick away. After that, you're going to place a cover slip onto the slide just like you did to the last onion one, and you're going to use low power to focus it. It says you'll probably not see the cells at this power, so you'll probably want to slowly increase the power to high until you can find an area with several good cells. So once you find a cell that you want to either draw or take a picture of, go ahead and do either of those in this section right here. And just like last time, you're going to label the cell membrane, the nucleus, the nuclear membrane, and the cytoplasm. Just like the last time, too, with onion cell, you're going to throw out the cover slip, not the slide, just the cover slip that you put on top of the slide. And then you're going to clean and return the slide so that the next class can use it. So after you've done your drawings or your pictures and you labeled everything, you're going to answer a few discussion questions in complete sentences. That's really important. So the first question is, how does the shape of the onion cells differ from that of the cheek cells? Which cells seem to be arranged in a more regular pattern? What structures were you able to see in both types of cells? Both plant cells and animal cells contain mitochondria, and yet they were not visible in the cells you viewed in this lab. Does this mean that these organelles are not found in the cheek and onion cells? Why or why not? Make sure to explain your reasoning. Last question. In class and in your reading, you learned that one difference between plant and animal cells is that plant cells contain chloroplasts. Were any chloroplasts visible in the onion cells? Why or why not? Again, explain your reasoning. When you're finished with your lab, go ahead and submit it to Schoology, and then when everyone's done, we will discuss it and go over it together. If you finish earlier than everyone, then you can go back to your seat and read your AR book until everybody's finished.